Polypeptide synthesis takes place in the cells of a living creature. It helps determine the function or role of each and every cell in an organism. The process can be broken down into transcription and translation. The first stage is transcription, and it takes place in a very secure nucleus that contains DNA which holds instructions on how to make proteins. This is what the DNA actually looks like. The nitrogenous bases are adenine, thymine, guanine, and cytosine. Anyway, back to protein synthesis. The DNA cannot leave the nucleus because of its size, hence it has no use. But wait, there is hope. Ribonucleotide triphosphates are formed in the nucleus and are very much attracted to the DNA. Thanks to a gas leak of the special enzyme named helicase, the DNA can now unwind. Here we see the ribonucleotide triphosphates attaching themselves onto the uncoiled DNA strand. The ribonucleotide triphosphate joins up to the DNA bases. This causes mRNA to be formed. mRNA is different, however, because thymine is now replaced with uracil. As the mRNA enters the cytoplasm world, his friend the ribosome shows up. From here on forward, everything takes place in the ribosome, and here we start the process of translation. To recap, mRNA is really important in the process of protein synthesis, because DNA in itself cannot leave the nucleus, and the mRNA can transfer information very easily. Translation is conducted in the cytoplasm and occurs when mRNA is processed by a ribosome. Ribosomes can be broken down into a large subunit and a small subunit. Ribosomes are made out of ribosomal RNA, and they contain tRNA and amino acids that are ready to make proteins. To simplify this process, imagine the tRNA being a builder, ready to make a house. Let's also imagine the amino acids being bricks. The only issue is, tRNA doesn't know what type of house to make, so he looks to the mRNA, which basically shows him the blueprints of the house. Now that you have a basic understanding of transcription, let's take a detailed tour of what's going on. So what you see here is the mRNA that has the instructions that would lead to the creation of the amino acids MET, cis, lisp, and asp. Once the tRNA hits the stop codon, it will stop adding to the polypeptide chain. As you saw with the animation, the tRNA leads the bases in sets of threes, or codons. As it went along, it interpreted those bases and found the corresponding amino acids. Then the tRNA released the amino acids and they formed a chain that gets coiled up to become a protein. To recap, mRNA is important in translation because it tells the tRNA how to make a specific protein, and tRNA is important because it physically moves the amino acids. Thank you for listening.